All right, welcome everyone. Today we have Yvon Ukwishaka who will be presenting on unraveling the intricate redox dynamics in inorganic nitrogen species in the vadose zone. So Yvon had his bachelor's in here through CASNA, where he majored in integrated science and also minored in water science, precision agriculture, and conservation agriculture. So we'll give him the space to present today. And also, let's remember that there will be a happy hour after now at the Lane Team pub. So, let's try to take advantage of that. Thank you.
Thank you so much. Does everyone hear me okay? All right. Thank you, Uche, for a good introduction. Yeah, so my name is Yvon Ukwishaka. I am a second-year master's in natural resources science. I work with Dr. Arinda Malakar in Malakar Critical Zone Research Lab. Before I begin my presentation, I would like to acknowledge the funding agencies who believed in our idea about nitrogen species. I would like to start with USDA, USGS, Nebraska Water Center, Water for Food, SNR, and University of Nebraska-Lincoln. They believed in our idea where we are going to talk about the intricate redox dynamics of inorganic nitrogen species in the vadose zone. In this seminar, you might hear the word inorganic nitrogen species and the vadose zone so many times. I am not expecting everybody to be familiar with those terms. So to break it down, we all know that nitrogen is just seventh element on the periodic table, but it's not only that it's just seventh element. It's the most abundant element or gas in the atmosphere. It's accounting 78% of the atmosphere. It's also critical nutrient for living organisms, animals and plants through many forms where you can find all these forms here. However, it's not accessible in gaseous forms. You can see all the gas forms here highlighted, and it might be accessible to plants through the common inorganic species, which are known to be just nitrate and ammonium. But how can all these forms be found in ecosystem? They can be found in ecosystem through different processes. As you can see from this picture is just showing what we call critical zone. Critical zone, it's low atmosphere all the way to the groundwater we have in our Earth. So we can see some of the processes that can happen, including atmospheric nitrogen fixation. We can find the one which we need to be worried about, which is just sourced from nitrate fertilizer, which is found mainly in agricultural intensified areas, including Nebraska. After having all these sources of nitrogen species, we might find that after these redox transformations or reduction oxidation transformation, they end up in just two different areas. Either they can be lost in atmosphere through denitrification or ammonia fertilization. They can also move down, keep moving down until it gets to the vadose zone and go to the groundwater system. Mainly, which one we should be worried about? It should be this nitrate. The vadose zone itself is the unsaturated zone between our land surface to the groundwater. It's very useful in water and contaminant filtration because everything, before it gets to groundwater, has to go through this porous medium, which is unsaturated. It's also critical for nutrient cycling, where you can find this nitrogen cycle, which is yet unknown in the vadose zone. Also, the carbon sequestration happens in the soil. Also, it's a storage for minerals, mining, diamonds, all these minerals that we need. Even though it's necessary and it's critical for our life, it's very hard to study. Because of the many reasons, few papers have discussed how it's hard, where they talk about the depth to water table. It can be 140 feet. No one is going just to go out in the winter or summer just to go to dig down 240 feet, but it may be also 10 feet or 3 feet. That happens. Also, it's very hard because it's a heterogeneous medium. You can find many things in there. As I mentioned, this is yet least explored part of the earth. You can find microbes. You can find in different movements that happens depending on the soil structure. Also, the weather is another challenge that we face when trying to understand the deep vadose zone. When it's winter, we don't go there. When it's too hot, nobody want to go there. That makes the groundwater monitoring a paradox, because the time you are going to detect the contaminant in your groundwater, meaning your vadose zone is already contaminated. So there's a high need to understand all the processes that happens in the vadose zone, which is hard again because of the reasons I mentioned above. However, we are not really going to be like, we are not going to just say we cannot do it. Let's now talk about the common inorganic nitrogen species, which I mentioned before to be nitrate and ammonia. How do they move? How do they transform within the vadose zone? From this good picture, it shows a small section of the critical zone where you can find this plant. I don't know if it's a soybean or corn, but let's assume it's a corn in Nebraska. So within this soil profile, you can see we have nitrate, ammonium, and soil particles. And we also have the precipitation happening. The component that we have here is just water and those common inorganic species. As you can see, nitrate, they have arrows showing from top as it gets down here. But for ammonium, they're going to stay here. The main reason is that nitrate, it's negatively charged. So are soil's particles. So they are going to repel to each other. But for ammonium, they have this like magnetic property. It's going to be stuck to the soil, yet it's going to be staying in there. So we should be really worried about nitrate, because it can just keep going to the groundwater, which is now serving more than 2.5 billion people on the planet as a drinking water. Moreover, 70% of food production industry depends on groundwater for irrigation. And then for this process to happen, we can find nitrate and ammonium moving or staying in the soil. It depends on the redox species available. There can be oxidizing agents. There can be reducing agents. Also, one more critical component is the vadose zone properties, either chemical, physical, or biological. Also, one more factor can be the microbes, the quantity, how much microbe we have there. What are the types and distinct communities we have in there? What are their functions in the transformation of those two inorganic nitrogen species? Looking at the global vadose zone nitrogen storage since 1925, I guess that's after Industrial Revolution when people started doing agriculture more and more. The nitrogen that we had over in that period of time, you can see that it's almost low. We had nothing. After 25 years in 1950, we found that in this region where we find US, and specifically you can find here the Ukwishaka, but you cannot see it right now, and sometimes some part of China and in Asia as well, there has been just increase in that nitrogen or nitrate stored in the vadose zone. And after another 25 years, that's 1975, we can clearly see that the storage shifted from low to medium. You can see that from here, you can even see that from here. Another 25 years later, that's in 2000, you can clearly see that this is where the Ukwishaka is sitting. You can see that it's moving up to high. This is concerning. Looking at here, again, higher vadose zone nitrogen storage in kilograms per hectare. We can imagine until today how much we have in there. So if it's stored and we keep doing the irrigation and all the industrialization keep going, people growing, we need more food as we apply fertilizer, mostly nitrogen containing. What's happening to our groundwater? Studies have gone through that, and they have found that we have nitrate in our groundwater system. As I mentioned, groundwater is serving more than 2.5 billion people for drinking water. From this map here, it's a study done by Nolan in 2006, where they developed this model to predict the nitrate concentration in the groundwater, either in shallow and in deeper aquifers. Here it's just 10-meter deep, which is considered to be the shallow aquifer. Here it's 50-meter deep. You can see that within shallow aquifer, again, we have this high concentration, which is exceeding 10 ppm, which is EPA's MCL, or maximum contaminant limit. That's the allowable concentration you can have in your drinking water. Again, that's just for shallow one, because as I mentioned, the vadose zone physical properties can impact how fast and how far your nitrate is going to be found at. If it's shallow, it's going to move so quick. If it's deeper, it's going to move so slow. It can take 10 years, 20 years, we don't know. But still, if you go deeper, you can still find some amount of nitrate in your aquifer, again, exceeding 10 ppm. Now the question is, how this agricultural intensification can impact our aquifer. From this map here, you can clearly see that as you apply the nitrogen, you are increasing the risk of groundwater contamination. And then your vulnerability of aquifer is also increasing. Looking at here, where Nebraska is sitting, heavily intensive agricultural state. All these locations here, they are sitting on top of agrarian aquifer. Not only that, you can also see here in California, where we have the Central Valley aquifer as well. So the more we practice agriculture, the more we put in more nitrogen in form of nitrate. The question is, should we drink our water? We don't know. Looking at the nitrate variation and irrigation status in the US, from this map here is from 1995 to 2015, where they looked into how many violations that have been happening in the entire United States. From the legend here, you can see the mean annual violation. The blue one, 0, where you can find these states. But if you go to red one, you can find between 37 to 63 violations each year. You can find it in California, Texas. Nebraska is not too far. It's between 24 and 36 and increasing. Why is that? Because again, irrigation as a main driver of the nitrate to our groundwater, as nitrate is soluble in water and moves so faster. You can see Nebraska is #1 state which irrigates more, 14.8 million acres in the US. Percent, that's about 8.6 million acres. One more thing we can also see from all these states where you can find so many violations, let's take a look at the county level of the irrigation status. If you look at Nebraska here, you can see that we have so many irrigation points across the United States, where each dot here equals to 10,000 acres. So this is high rate of irrigation that's going on. If we irrigate, meaning we also supply more nitrate containing fertilizers. Also, one more driver for this number of aeration includes the change in irrigation systems. These irrigation systems were since 1984 to 2018, there's been change from the gravity system moving to pressurized system. That includes pivot irrigation system right now. So this change here doesn't mean the nitrate which has been in the soil is going anywhere. We are pumping still more nitrate. Even though we can change the irrigation type, we are still pumping more water and more nitrate. Again, all of this is happening in agriculture intensified areas. don't drink water that have high concentration of nitrates and ammonia or nitrites, which are the same forms of nitrate. The main reason is that you all have heard about blue baby syndrome. That's a disease due to consumption of high nitrate concentration. Also, some recent studies that have shown that nitrate concentrations has been linked to some type of cancer. For children and pregnant women, they may suffer this one disease. It's anoxia. It's like internal suffocation. We shouldn't drink this. We also like that in Nebraska, there have been vadose zone study aiming to understand nitrate status in our vadose zone. From this map here, shows the red points. Those are the locations where there have been previous vadose zone studies, at least dating back to 40 years ago, even more. There has been intensive study on nitrate in Nebraska. So if you visit this website, you can find all the data about this one. I would like to highlight this one study that came out in 2018 from Adam. It was his thesis, where he was looking into comparing how is the inorganic nitrogen species under two different irrigation types, gravity and pivot irrigation systems. By USDA, gravity irrigation, they use more water. But pivot uses almost between 62 to 70% less water compared to gravity. What they found was that in residential area where they don't apply much water, it's in between, it's like they receive just rainwater and you can just irrigate your flowers, your vegetables, you have some. But they found that on the nitrate side, the residential area, they have significantly less amount of nitrate in the vadose zone. But if you look at between gravity and pivot, look at the gravity here. High amount of nitrate because you are pushing more water in the soil than it goes with the nitrate we have. Looking at the ammonium side, it makes sense that where we have high nitrate, we are going to have, it makes sense that where we have high nitrate, we are going to find less ammonium. So look at this one. For a pivot irrigated area, we have high ammonium concentration. Less water, then your nitrate is going to stick with the soil, then be used up by the plants. So this study ended up concluding that the inorganic nitrogen transformation is a function of irrigation practices. One more study that came out from my research group came out this year. They looked into also how pivot and gravity irrigation can influence the redox chemistry of the vadose zone of nitrogen. They found that in gravity irrigation, you have high nitrate and high ammonium and high water. And the pivot, low water, low nitrate moving into your soil. And then from this bar graph, you can find that under gravity irrigation, you have significantly high amount of nitrate compared to pivot. On the other hand, in the gravity, we have significantly less amount of ammonium compared to pivot. Now, points to ponder, we can find out irrigation and fertilizer application influence the vadose zone geochemistry. We can also note that inorganic nitrogen transformation is a function of irrigation practices, as we saw from previous slide. Also, the vadose zone is a heterogeneous medium. We have so many things going on, and it scares me because you cannot cover the full range of what activities happening in there. From there, we came up with the hypothesis that irrigation practices beneath agricultural lands drive inorganic nitrogen species transformation in the vadose zone. Now the question is, how can we appropriately unravel the intricate dynamics of reactive nitrogen species transformation in the vadose zone? What do we need? We need to recreate undisturbed vadose zone in a column in a controlled setup, where we can just control what's happening there, so we can eliminate some of those variables we are not interested in. After that, we need to evaluate the reactive nitrogen redox-based impact on, here is the type of uranium vaporization, should be nitrogen transformation at the different zones within the vadose zone. We can start with root zone, which is highly reactive. We can go also to capillary fringe, which is the attachment to the groundwater. And then see how the redox chemistry is changing. Unlike the previous studies, here's the keyword, undisturbed. Due to the challenges associated with understanding the deep vadose zone, some of the previous studies, they took the soil cores, they disturbed them, repacked them in columns. The issue with that is that they are just disrupting physical, chemical, and biological properties of the soil. To capture the full range of that, we need to use undisturbed soil. In our experiment, we selected a location. It's a Bradshaw. It's in York County, an hour from here. The main reason for this location is that previous studies have showed that there is an increase in nitrate concentration in the drinking water. Advantage we had was that it has a nearby well where you can just collect fresh groundwater, which we used in this project. Also, we had a weather station. Since we are creating the entire vadose zone, it's like taking the field, bring it to the lab, and evaluate the redox chemistry. We knew that the soil was well drained. We knew the irrigation history of the field, which was pivot, and it was previously corn-planted. Then we came up with a unique design. From the location, this is a picture of a geoprobe. It's a machine that we use to drill down and get the fresh soil, which is undisturbed. From the selected location, we collected two 21-meter deep intact cores. We brought those to the lab. The first thing we did was to characterize them, to know what kind of textures we have in there. And then we repacked them into 160 centimeter columns, which are plastic and visible. So we can see how the water is moving. And if we can have algae growing there, we can know something is going on. How did we do it? From those two deep cores, we packed them into two systems. Each system had 3 replicates. One system was irrigated, another system was rainfed, because we're looking into how irrigation amount is going to influence the nitrogen transformation in the vadose zone. From each of the liners, so if we have, for example, 21 meter deep cores, we are going to have small sections marked by depth. From each of the liner, we took small segments, repacked them into our 160 columns. We packed them by mix and match. We also made sure that we have one variable. We didn't supply any external nitrogen, either in form of fertilizer or from the groundwater. We also didn't plant any corn on top. That's why we didn't supply any fertilizer. But to make sure we have food for microbes, we supplied root exudate, which is the juice produced by roots of any plant, in this case of corn, so that microbes can get food, as microbes get food from carbon. Then after repacking our columns, we had our area of interest. As I mentioned, we are looking to root zone and capillary fringe. But since all these zones can have implication, negative or positive implications, we also want to measure what's going on with our groundwater system. So we had also to monitor the groundwater. From that, we used Rhizome semperas. These are very tiny tubes, very porous tubes, 0.2 microns for size, which are well known to get pore water samples from undisturbed soil. So we made small openings at root zone, which is just 10 centimeters below the surface, and capillary fringe, which is 10 centimeter from water table. And then we had to add one in the groundwater system. From there, that's where we collected biweekly water samples. We ran this experiment for 159 days. So the gap you can see here before we took our first sample was to condition the columns. We need to give the columns enough time to be at the field level. That's when we start taking samples. Before we plant anything, of course, rain is going to happen. Here, this deep dark blue bars, they're showing artificial rainwater, which was supplied as it was happening out in the field, because we had an advantage that where we collected soil cores, there was a weather station within 5 miles radius. And then from the well, which was 1 mile from the collected location, we collected fresh groundwater sample, which were used for irrigation. The irrigation was supplied from day 97 until day 107. It was done just once because we couldn't find any information about irrigation scheduling from the producer. But we had to make sure that we supply irrigation to see the impact. And of all 6 columns I mentioned, which were divided into 2 systems, all of 6, they received artificial rainwater regardless. But for our treatment, which is irrigated, we supplied we supplied irrigation water to see the impact. And then from day 61 until day 159, we collected samples biweekly and analyzed them for multiple ions in the soil. From the questions we wanted to ask, we want to see, do nitrogen species change in the vadose zone sediments? By looking at the nitrate in our soils, the black one is showing day 0, which is the pre-soil, the soil we took out in the field to look into how nitrate concentration is. And then red one is irrigated, blue one is rainfed. From this graph, it's just the concentration on X and depth on Y-axis. From what I observed is that we had high concentration of nitrate at the root zone, from at least 40 up to top surface. We had high amount of nitrate, but the rest, there was no significant difference. The same goes for sediment ammonium, where we have, again, from 60 up to top. For some reasons, we had this variation. For this one, it makes sense because from here, if we had this amount of nitrate and we supply water, we are going to wash away nitrate and it goes down. For here, in a rain-fed system, we had almost 3 times the amount of ammonium we had initially. There is a mechanism that happened here, which might be converting our ammonium into our inorganic ammonium into organic ammonium. We are yet to find out because we are still running TKN, which is going to give us organic nitrogen species and ammonia in our samples. One more question. Nitrogen species in the vadose zone pore water. As I mentioned, for the entire period of time, since day 61 until day 159, we collected biweekly samples, pore water samples from the locations I mentioned, root zone, capillary fringe, and the groundwater. What was found is that this graph is showing the average of the entire period. What we observed was that where we applied much water, that's in irrigated system, where we supplied both rainwater and irrigation water, we found that there is a significantly high amount of nitrate compared to rain-fed. The same trend was observed across the entire vadose zone. where we found again at the capillary fringe, and the groundwater system, there was no significant difference. On the ammonium side, it's vice versa. As I mentioned, where you have high nitrate, you are going to find less ammonium. So here in our irrigated system, we have less amount of ammonium, and in a rain-fed system, high amount of ammonium. The same trend followed. Again, the mechanism behind this, we are going to go into it afterwards. To remind you, nitrate is oxidized form of nitrogen, and ammonium is reduced form of nitrogen. Meaning, to have nitrate this high, there has to be oxidation, or there has to be some mechanism, either redox mediated or microbiome mediated. From there, we can also conclude that nitrogen transformation is a function of irrigation practices, as we've seen here between irrigated system, which received extra amount of water, and rain-fed, which received just precipitation. Let's now dive deep into step by step how we can signify that more amount can induce high nitrate. Let's look into the day-wise nitrate concentration in pond water. Again, from day 61 up to day 159. All of them, when they're receiving just precipitation, they had the same, almost the same concentration. Looking at the black one, which is the red zone, which is the high reactive zone in our system. But after we supplied here, after we supplied the irrigation amount, look at the spike here. This burst that happened here came after we supplied more water. It's being observed in irrigated, but in rain-fed, nothing happened. This is the day, this is the irrigation event that happened. This is the spike. So we can say, from this one, we can say that if we supply more amount of water, we are going to find high amount of nitrate. It's not always the case, but it's possible. To confirm this, we had to look into the redox process indicators, because if you do nitrate and ammonia, you're not going to just come up with a mechanism. We have to measure some indicators that can be like, there has been oxidation, there has been reduction, as this is redox-based process. So from this table here, we measured reduced iron. We measured ORP, oxidation reduction potential. We measured the pH, which is another great factor, especially when you are considering microbe-mediated transformation for both irrigated and rain-fed. Then we have to look into which zone. From this picture here from Bosch, this is the redox ladder. It can predict and tell you which zone you are in. By looking at the average, the pH in our irrigated system ranged between 5.9 to 7.7. If you look, it's around here. You can look into nitrate can become nitrite. Manganese can change the oxidation state. But this one is not enough to give us all the details we need to know. That's why more tests are needed. We need to know if there's a conversion from inorganic species to organic species. We also need to know, is it really redox-based or there's also microbes involved? We are still waiting for the data. So from what we have seen so far, the probable mechanism can be explained by this graph here, which is showing dissolved oxygen, and oxygen is the #1 oxidizing agent we know. So we took a measurement of the capillary fringe of the dissolved oxygen. What was found is that the black one is showing the irrigated and red one for rain-fed. So after we supplied the irrigation amount, like a week after, we have seen the spike in oxygen dissolved in the groundwater system. Meaning, as we supply water here on top of our columns, it's pushing down the oxygen all the way down there. So as oxygen is moving from here to down there, it's causing oxidation. That's why we have seen that nitrate on the root zone way higher, and the trend kept going. This is the probable mechanism. To end, I would like to thank USDA NIFA pre-funding, which is now supporting the rest of the test. We are running right now, the TKN, even microbiome, where we're looking to how much DNA or microbes we have there and what are they, and to come up with their specific function in the redox process or nitrogen transformation. I also like to thank Water Sciences Lab, where we did all the chemical tests. I would like to thank our collaborators, Dr. Dan Snow, Michael Kaiser, Dan Miller, and Dr. Ray. Also, the funding agencies, USDA, USGS, NWC, Water for Food, SNR, and UNL. Thank you so much, and any question?
Thank you so much, Vaughn. Are there questions? So if there are questions for Vaughn. All right. Okay.
Yeah. Yes. Well, that's a good question. With the climate change, when there is low frequency of rain, people tend to irrigate more. Still, the soil is going to get the water it needs. So this might change. It might be happening just at this one specific location we selected. But more researchers are needed. People need to keep doing this. This was just 159 study, where we had about, I don't remember exactly how much rain we received, but when it's a drier, you cannot find this. So that's where people need to work together, people in irrigation field, people in climate field, to come up with a good way to manage the water resource. But from what I can tell, not only this study, also the two studies that I have talked about in here, they have found that more water, more nitrate going in our soil. The end goal is to find just this one technology where you can be like, okay, if you apply this amount, you are not going to cause any problem. Or if you can find this something which can convert nitrate into less harmful chemical, that would be great. So more research is still needed to see the variation between where you have too much rain, too less precipitation. Well, about nutrient management, people prefer different ways. People do cover crops. People do no-till, do conservation tillage, or all the other methods. About that, it's a different case. From the field where we took our sample, it was no-till, and they haven't had cover crops for at least past 5 years. So I will look into where they did the same study, where they had different nutrient management practices, and see how the nitrate looks like. Yeah, those are good questions, actually. Thank you.
So I'm interested in so I want to know if you talk about the soil characteristics, especially if you have something like this for that you need to rather from that.
Well, we did. So again, as I mentioned, before we packed our columns, we had to characterize soil and see what are the soil textures we had in there. The infiltration was very easy in this experiment because we didn't have high amount of clay. But if we have clay, so for any contaminant or water to move beneath any zone, we have to account for the water holding capacity of that zone. If it's going to hold more water, the nitrate is going to stay there, or any contaminant. Because those contaminants, they move with water. That's the only way. But if you have sandy soil, it will be very easy. So the water is going to keep going. So also we need to look into, as I mentioned, for these contaminants to go, we have to look into the redox species we have in there. We have to look into soil or sediment physical properties, chemical properties, biological properties. All those, the combination of all those factors, they are going to influence how your chemical or how your water is going to move in the vadose zone.
All right, thank you.